Hi, today we're going to talk about the structure of DNA. And I know that sounds a little dry, but we really need to understand how DNA is put together if we're going to understand things like how DNA is replicated or how messenger RNA is transcribed from DNA. So these are the questions we're going to answer today. We're going to answer, what is a nucleotide of DNA? What does that mean? We're also going to talk about, well, how do nucleotides relate to nucleic acids? And we're going to talk about which nucleotides are found in DNA. And we're also going to talk about, well, what does it mean when we say 5' or 3' ends of DNA? What does that mean? So I think a good thing to do might be to start a separate list of all of this different terminology that we're going to learn today, because biology is full of jargon, because we have to name these things certain things. And the more you understand the terminology, the better you're going to be able to understand the structure and how DNA works. Now, I want to remind everyone about the flow of information in cells. Remember, DNA to RNA to protein. And this is called the central dogma of molecular biology. Now, DNA contains all of the hereditary information for living things. So all living things have DNA in their cells. DNA is what's passed down from generation to generation, but it's also the instructions for how a cell lives. DNA is an abbreviation for deoxyribonucleic acid. Now, I'm going to pause here because I want you to say that word, okay? Become familiar. We have to say these things so we can become, familiar with them. So deoxyribonucleic acid. Deoxyribonucleic acid. Exactly. That's what DNA stands for. And we're going to talk about later today why it's called deoxyribonucleic acid, because that's a mouthful. Now, DNA is used as a template to transcribe RNA. And RNA, right, is copied from DNA. So RNA is a temporary copy of this information. RNA stands for ribonucleic acid. Okay, so say that term. Ribonucleic acid, exactly, ribonucleic acid. And then RNA is read by ribosomes, and the ribosomes use the information in that RNA to assemble a protein. Okay, so RNA carries the instructions for making proteins, and those proteins are going to be made out of amino acids, and it's the ribosomes within the cell that make those proteins. So let's talk about DNA, right? DNA is a nucleic acid. That's what the N and the A stand for. If you remember, deoxyribonucleic acid, deoxyribonucleic acid. So the N and the A stand for nucleic acid. And what is a nucleic acid? Well, nucleic acids are long strands of nucleotides joined together. So if we think about, making DNA, it's not just one long structure that starts at one end and then the molecule is made and continues being created, right? It's actually smaller pieces that get joined together. And those smaller pieces are called nucleotides. So DNA is actually composed of over, in humans, over 3 billion nucleotides. Of course, smaller organisms, let's say E. coli, a bacterium, it has more like in the millions of nucleotides joined together. But in humans, it's over 3 billion nucleotides that create our DNA. And DNA is a nucleic acid. So the nucleotides are the smaller pieces that get bonded together to create the bigger structure. And the bigger structure is called a nucleic acid. So you can kind of think of it like a train, right? If you're sitting at a train track and a train goes by, the whole thing is the train. And it can be really long sometimes. But that train is actually made of individual railroad cars, right? And those railroad cars get joined together to create the longer structure, which is the train. So in this analogy, those individual railroad cars are like the nucleotides. They get joined together to form the entire long structure, and that's the DNA, the deoxyribonucleic acid. Okay, so this is a graphic of DNA, and we're used to seeing it. It's this helix. In fact, it's a double helix because there are two strands in here, right? We can see here's one strand that I'm outlining with the mouse. And then there's actually another strand over here. Okay, so it's a double helix because these wind into this spiral-like structure that we call a helix. But that's the whole thing we're looking at there. In this cartoon, we're looking at a piece of DNA, and there's actually many nucleotides that are joined together in this piece of DNA. So it's really important to understand what a nucleotide is. It's that basic building block, right? Like in our train, it's an individual railroad car, okay? So in a piece of DNA, it's made up of nucleotides. And this is a cartoon I put together just to sort of show you what's found in a nucleotide. And in a nucleotide, okay, we have a phosphate group, and that phosphate group is attached to a sugar that has 5 carbons. And then on the other side of the sugar is bonded what we call a nitrogenous base, which sometimes you'll hear is called a nucleobase or just a base. So this is the structure of a nucleotide. Now, just as a reminder, a phosphate group is a phosphorus atom that's bonded to 4 oxygen molecules. Okay, you can see 3 of those are with a single bond, and 1 of the oxygens is bonded to the phosphorus with a double bond. So that's a phosphate group, okay? Now, again, this is the nucleotide we're looking at. The phosphate group is bonded to a 5-carbon sugar. And so sugars have carbon, hydrogen, and oxygen in them. And in this case, the sugar is known as deoxyribose. Now, maybe let's think of some other sugars, names of sugars that you've heard of. Maybe sucrose, like table sugar, or glucose, or fructose, like high fructose corn syrup, right? What you might notice about sucrose, glucose, fructose is they all end in OSE. Sugars end in OSE. And so that's a reminder for us. This deoxyribose sounds very chemically, but it ends in OSE. So we know it's a sugar, and it has 5 carbons in it. Let's say glucose, for instance, has 6 carbons. Deoxyribose has 5 carbons. And this is where the name deoxyribonucleic acid comes from, because DNA has the sugar deoxyribose in it. Now, just as a side note, when they put the whole thing together, just to make it flow better, they dropped the S and the E from deoxyribonucleic acid. Okay, I guess it would have been a little harder to say deoxyribose nucleic acid. So they dropped the S and the E so it flows better. Deoxyribonucleic acid, and it's called that because the sugar found in the nucleotides of DNA is deoxyribose. Now, on the other side of that sugar is a base, and it's called a nitrogenous base, or like I said, you might just hear nucleobase or base. But I like to say nitrogenous base because it reminds you that we're going to find a lot of nitrogens in it. So this is a nucleotide. And again, in human DNA, we're going to have billions of these nucleotides that become bonded together to form our nucleic acid, our DNA. This is a nucleotide. Now, here is our nucleotide. And there are 4 different bases that can be found. So we actually have 4 different flavors of nucleotides that are found in DNA. And the difference between those nucleotides is the type of the base that's there. So all 4 are going to have that phosphate group bonded to the sugar, deoxyribose, and then it can be bonded to one of 4 different bases. And you're probably going to recognize these: adenine, guanine, cytosine, and thymine. Those are the 4 different bases that can be found in the nucleotides of DNA. So again, when you hear something like guanine, well, guanine isn't the entire nucleotide. Guanine is the base that's found in the nucleotide. Okay, so when we're talking about adenine, guanine, cytosine, and thymine, A, G, C, and T, as we like to abbreviate them, we're really talking about the bases that are found. And there's only one base per nucleotide, but there are 4 different flavors, 4 different types of nucleotides, because we can have nucleotides that have adenine as the base, guanine as the base, cytosine as the base, or thymine as the base. So this is the general structure of a nucleotide. So we have our phosphate group, our 5-carbon sugar, which we know is deoxyribose, and we have our one of 4 different nitrogenous bases that are found there. Now, this is just the line structure of this is just the structural drawing of a nucleotide. I didn't want to throw this right at you because it looks like a lot of chemistry, right? Which can be intimidating. I absolutely get that. And that's why I gave you sort of that little drawing here on the side. But now that we understand the different parts of it, we certainly can like break it down and understand how this works, right? So here's our phosphate group right here. We know that's going to be the phosphorus atom bonded to 4 oxygens. And then this part in the center, that's deoxyribose. That is the structure of the sugar that's found in a DNA nucleotide. And I know you're thinking like, wait, you said there were 5 carbons in sugar, in deoxyribose. Like, where are the carbons here? It doesn't look like there's any carbons. And well, when we're using these structural drawings in chemistry or these line bond drawings, they remove the carbons. So just to make it look easier, and most of the hydrogens too are removed because there's lots of hydrogens in here as well. So anytime you really see a crick or you see like a little bend in the molecule, there's going to be a carbon there. It's just that they don't put it in because there's lots of carbons here and it would get sort of intimidating to look at. So we can count those different carbons. Okay, here's 1 carbon is here, 2 is here, 3 is here at this bend. The next bend is 4 carbons, and then the 5th carbon is actually sticking up over here. Okay, that's the 5th carbon, and that's where the phosphate group binds, is at the 5th carbon of this sugar. Okay, so even though they're not drawn here, there is a carbon here, 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 and here. Those are our 5 carbons. And you notice there's an oxygen here as well at the top of the deoxyribose molecule. Now at that first carbon right here, that's where the base gets bonded into the nucleotide. And so we can see this base here, right? We see it's, just generally looking at a structure, what do we see? We see there's some single bonds, some double bonds. It's made up of 2 rings of different atoms here. And of course, as promised, it's a nitrogenous base. So you see all those ends. There's a lot of nitrogen in this base, in the bases, and that's why we call them nitrogenous bases. So that's sort of breaking down the structure of a nucleotide and going into a little bit more detail about what we see chemically in the different parts. And why do we care? Well, to really understand how these nucleotides get bonded together to create the big structure, the big molecule of the nucleic acid, we sort of have to understand the different parts of the nucleotide. Now, we talked about how there are 4 different nitrogenous bases, 4 different bases that are found in DNA. Again, here's our nucleotide cartoon over on the right, and we're talking about the base, the part all the way on the right of this drawing. There are 4 different types of nitrogenous bases that can be found in DNA. And maybe you've heard of them, right? Cytosine, thymine, guanine, and adenine. These are the different bases that are found in DNA nucleotides. And we're not going to memorize the structures here. We're simplifying it today. We're not going into all of the chemical details necessarily. But if you just take a step back and you look at these bases, what do you notice about them? What do you notice about the structure of them? Yeah, maybe you're like, well, cytosine and thymine, they're made up of 1 ring structure. And guanine and adenine, they actually have 2 rings that are stuck together in their structures. And yeah, we see nitrogen in all of these molecules. So nitrogenous bases, they have nitrogen in them as well. And we can categorize them into sort of different kinds of groups based on their structure. Now, cytosine and thymine, they're made up of one ring only. Okay, you can see that the atoms form a ring structure. And so cytosine and thymine are derived from what we call pyrimidines. So we call these the pyrimidines. And again, that just means it's a structure that has, a single ring to it. Now, guanine and adenine, they have two different rings bonded together within those bases. So those are called purines. Now, again, why do we care about all these details when we're talking about DNA nucleotides? Well, later on, we're going to find that when the bases pair with each other from one strand to the other strand of DNA, it's going to be a pyrimidine that pairs with a purine. And just as a little preview, cytosine is always going to pair with guanine, and thymine is always going to base pair with adenine. So we sort of keep this in mind to really understand which ones pair with each other and how that's going to work in a structure of DNA. We're not going to talk about it today, but RNA has an additional base other than what's seen here. Instead of thymine, RNA is actually going to have a base called uracil. So in DNA, we have cytosine, thymine, guanine, and adenine. But in RNA, we have cytosine, uracil, guanine, and adenine. And I mention this just so you're aware of it, but also I have lots of little mnemonics that help me remember these things, and maybe they might help you too, even though they're a little bit silly. And I remember that thymine looks a lot like uracil. So uracil is another pyrimidine. So our pyrimidines in nucleic acids are going to be cytosine, thymine, and uracil. And the word pyrimidine looks a lot like a pyramid. And so in my head, I remember that pyramids are cut from big stones, okay? C-U-T, cut from big stones. And C, U, and T are the three pyrimidines that we find in nucleic acids, okay, nucleotides. So cytosine, uracil, and thymine, those are the pyrimidines. So that leaves the other two, guanine and adenine, to be the purines. That's how I remember that and thought I'd share just in case it can help you as well. So these are the four different bases that are found in DNA: cytosine, guanine, adenine, and thymine. Now remember, here is our cartoon of a nucleotide. And if we switch that out with the structural drawing of that nucleotide, we see that there is one of those bases that is in this particular structural drawing. Now, looking at this base, right away, you see it has two rings. So you know it's a purine rather than a pyrimidine. And we know our purines are guanine and adenine. So can you identify in this structural drawing of a nucleotide which of the bases is found in this one? Okay, well, let's see. Looking over here, we see a lot of nitrogens in adenine, a lot of nitrogens in guanine. Okay, but looking at the different structure here, well, there's a nitrogen coming off of this second loop here. Okay, in adenine, there's an oxygen that comes off from that ring structure here. So in guanine. So if we look over at our structure, well, it's the nitrogen that is coming from this second ring. And so in this case, this particular base found in this nucleotide that we're looking at is adenine. See how that all comes together? Now let's talk about how we assemble DNA. Like I said, human DNA in our cells is made of over 3 billion of these individual nucleotides. So how do they actually join together? And the answer is the DNA is composed of strands of the nucleotides, and the nucleotides get bonded together. They bond together with a covalent bond. Officially, we call that bond that bonds the nucleotides together, we call it a phosphodiester bond. And I just mentioned that in case you've heard it before and you want to know what we're talking about. It's a covalent bond, and that's the bond that binds nucleotides together, a phosphodiester bond. Now, not only does DNA have one strand of these nucleotides bonded together, it has a second strand right? And those strands are going to connect together. The bases are going to also bond together through hydrogen bonds. So we have the two strands, the bases are going to fit into the center, and then that whole thing twists into a helix, a double helix, because there's two strands that are twisting into that helix. So if you think about it, DNA sort of resembles a ladder if you sort of unwind it so that it's flat. And on the sides, the sides of the ladders are going to be the phosphate, sugar, phosphate, sugar, phosphate, sugar. And the same thing on the other side. The other side of the ladder is going to be the phosphate group bond to the sugar, bond to the next phosphate group, bond to the sugar, phosphate group. So those sides of our DNA ladder, those are the two strands coming together. We call those the sugar phosphate backbone. And it's just like our backbone, right? Our backbone helps to keep our structure, and other things come off of that backbone. And it's the same thing here, where the sides of that ladder are made up of sugars and phosphate. That's the backbone, because then the bases are going to stick into the middle, and those sort of create like the rungs of the ladder. So I'm going to draw out how these different nucleotides are attached to each other, okay? Where does that bonding occur? So here we have one nucleotide, and you're going to have to excuse me because my drawing isn't so great. So bear with me, okay? My drawing skills, well, I should take a class someday. Let's just say that. So here's a phosphate group. And that phosphate group is attached to our 5-carbon sugar, which I'm going to draw the phosphate group as like a circle. And then the deoxyribose, the sugar it's attached to, I'm going to draw that like a pentagon. Now, we also have to put the base in. So the sugar is also attached to our nitrogenous base, AgCRT. And I'm just going to put a big rectangle there, okay? Because just to illustrate, it could be one of those 4. Now, here's our nucleotide. And now our next nucleotide is going to be bonded to this. So what happens is that, okay, we're going to have a nucleotide like this, phosphate group sugar, a nitrogenous base. The next nucleotide actually attaches to the sugar. Okay, so here's the phosphate group that attaches to the sugar. And then this phosphate group is part of the next nucleotide. So it has its own sugar. So here we are drawing in the deoxyribose. Okay, that's the next nucleotide sugar. And here's our next base. Okay, so we have one base here, and we have one base here that have been attached together. Now we have 2 nucleotides in our DNA. And the process continues so that the next nucleotide is again going to attach to the sugar. Here's our next nucleotide, our next deoxyribose sugar, and our next base. And this goes on and on and on until the entire base of DNA is made. So there we go. Here are 4 nucleotides that have been bonded together. Now, the part that has the phosphorus and the sugars, that's our sugar phosphate backbone, okay? And the bases stick out from that. So again, if we're sort of thinking of this as a ladder, we have our sugars and phosphates that create sort of the side of the ladder, and then the bases stick into the middle of it. Those are sort of like the rungs of the ladder. And that's half of the DNA structure right now there, because we know that it's double-stranded. There's another strand on the other side that's going to form the other half of the structure, or the other half of the ladder. So let's just put in a couple nucleotides of that as well. So here's phosphate group attached to our deoxyribose sugar, and then the base. There it is. Now, the next phosphate group attaches to the sugar. So phosphate group, sugar, and then base. And then the next nucleotide, here's phosphate group. Here's the sugar of the next nucleotide, and then the base. And we can go on and on and on. That's the other strand of the DNA. So what did you notice about how I drew it? Yeah, I drew it upside down. And that's because that strand is actually going in the opposite direction. So in our latter situation, if this strand was from top to bottom, going from top to bottom, then the other strand actually gets made from bottom to top. Okay, so in this case, we had our sugar phosphate backbone being made from bottom to top, and then the bases of this strand stick into the center. And the different bases, the bases that are sticking into out of the DNA backbone, sorry, the sugar phosphate backbone of one strand of DNA, those bases are going to bond to the bases that are sticking out of the other strand's sugar phosphate backbone. Now that's a hydrogen bond. So hydrogen bonds are really important, especially in biology, because they help to hold molecules together, but it is a relatively weak bond. So it's not like a covalent bond that's very difficult to break. Hydrogen bonds can be broken, and that's why those two strands are held together by hydrogen bonds between the different bases on one strand and the other strand. But when DNA needs to separate, those hydrogen bonds get broken. So there are these hydrogen bonds that are going to bond together the bases of one strand to the bases of the other. And there we are. There is our double, our two-stranded DNA molecule made up of nucleotides. And this is how the nucleotides are joined together, okay? So we have a phosphate group and a sugar. The base is going to stick into the center of the DNA molecule, and the next nucleotide's phosphate group, sugar, binds to the deoxyribose, the sugar of the first base. So we have phosphate group sugar, phosphate group sugar, phosphate group sugar, right? And then that's forming our sugar phosphate backbone. And then the bases are going to stick into the middle. And that happens with both of those strands of DNA. So we have DNA, we know it's composed of these two strands of nucleotides, but they're going in opposite directions. And that's called antiparallel. So to fit properly, for those bases to fit properly, one strand is going in one direction, the other strand is going in the other direction. So something that's parallel, right? If we have two lines that are parallel, it means they're running along each other at the same distance from each other. But the word antiparallel just indicates that one of those lines is going in the opposite direction, okay? Antiparallel. And it has to be that way for the nucleotides to fit properly together with each other. Now, just as a reminder, we talked about how one of those bases that sticks in the center is going to be a purine, and the purine base is going to bond to a pyrimidine base, right? And that helps us fit it together as well, because remember, the pyrimidines have one ring, but the purines have two rings. So if we had two purines together, it would sort of cause the DNA to sort of bulge out at that point. So whenever those base pairs are made, one is a purine and one's a pyrimidine. And very specifically, it can't just be any purine and any pyrimidine. It's always adenine and thymine, and then cytosine and guanine that are going to pair with each other. So here's our, our cartoon of DNA, our double helix, and we can see here's one strand of it, starts up here, right? We can follow it. There's the one strand of our DNA. And then there's a second strand as well. Here's the other side, the other strand of DNA that's formed like this. And we see the blue part here, this like ribbon that's made, that's illustrating that sugar phosphate backbone. That's made up of sugars and phosphates. And what's sticking into the middle here, these different colors, those illustrate our four different bases: adenine, guanine, cytosine, and thymine. And we see that the base on one strand, right, forms a bond, a hydrogen bond with the base that's sticking off the other strand, the nucleotide on the other strand. So parallel, right, anti-parallel. One strand is going one direction, the other strand is going in the opposite direction. And I kind of think of it like roads, right? If you drive on a road, and this is an American road, so forward is going to be on the right lane, on the right side of the road. That's forward. So if this lane is going in this direction, we know that the lane on the other side of the road is going in the opposite direction. This is actually anti-parallel. And DNA is the same way, that one strand goes in the forward direction this way, and the other strand goes in the forward direction in the opposite way. So they're sort of going by each other in opposite ways. Now, there's a way that we term the forward direction of DNA. And we use what's called 5' and 3' to indicate the direction of DNA. So 5' to 3', that's the forward direction of DNA. Okay, so if this is the 5' end over here, then we know 5' to 3' that this DNA strand is going from left to right. Now, what does that mean about the other strand going in the opposite direction? Well, forward is always 5' to 3'. This is the convention, and I'm going to explain to you why we call it 5' and 3'. But if this bottom strand is going left to right, 5' to 3', that means that the top strand of DNA, its 5' to 3' is going in the opposite direction. Okay, so DNA, the forward direction is always 5' to 3'. And this bottom strand here, this part of the road, is going forward from left to right, so 5' to 3'. The other strand of the road, of the DNA here, well, it's going forward from right to left, which is going to be 5' on the right to 3' on the left. So this is the convention that we use to discuss the direction that the DNA strand is going. And again, why do we care about this? It seems kind of like in depth, right? Why do we need to know this? We need to know this because we're going to refer to these directions when we're talking about how DNA is copied or how messenger RNA is made. Because new DNA is always put down in the forward direction from 5' to 3'. So let's look at a strand of DNA I made here, right? So in this graphic, this is the blue and yellow spheres here. This illustrates the sugar phosphate backbone. And then we have the different bases that sort of stick into the middle of our DNA structure. Now, this bottom strand, okay, here's the 5' end, and here's the 3' end of our strand of DNA. So let me ask you, which direction is the bottom strand of DNA going? Is it going left to right, or is it going right to left? Okay, so how do we answer this? We look at this and we say, okay, we know the forward direction is 5' to 3' always. So in this case, 5' is on the left, 3' is on the right, so that means that it's going from left to right. Now, just when we figure that out, we automatically know the direction of the other strand, because if this bottom strand is going 5' to 3' left to right, then it means the top strand is going 5' to 3' right to left. So if ever we take a double-stranded piece of DNA and we know, okay, on this bottom strand, that's the 5' end, we know that the other strand at that point is going to be the 3' end, right? Because they're going in opposite directions. So if this were, to be a piece of DNA and we were to add another nucleotide to this after this C, new nucleotides are going to get added to the 3' direction, because that's the forward direction. From 5' is the beginning, and then nucleotides are added towards the 3' end. So in this example, T would have been the first nucleotide, then A, then C, then G, then A, T, G, T, C, right? We're going from left to right. So 5' is the first one, and then the direction that it's going is towards the 3' end. So this is how we can say, okay, we know DNA is antiparallel, that one strand is going in one direction and the other strand is going the opposite direction. And the way that we label those is 5' to 3' is the forward direction always. Okay, so if this bottom strand is going 5' to 3' this way, then the top strand is going 5' to 3' in the other direction, because they're antiparallel. So let's take a look now at sort of, it's much prettier than my drawing, a graphic here of the different nucleotides that have been bonded together into a piece of DNA. So here, for instance, is one nucleotide, and you can see the phosphate group, right, which is bonded to the sugar deoxyribose. And here are the 5 carbons, 1, 2, 3, 4, 5. And then at that first carbon, that's where the nitrogenous base gets bonded. So in this case, it's adenine. Here's the next nucleotide, right? So we have in the first nucleotide, phosphate group, sugar. And then the next nucleotide comes, phosphate group, sugar. And we go on from there, phosphate group, sugar, phosphate group, sugar. So the phosphate groups and the sugars of each nucleotide get bonded to the next phosphate group and sugar of the next nucleotide. And that forms our sugar phosphate backbone. So you can kind of think of it as, 5' to 3'. On that 3' end, that's sort of the bottom end of the nucleotide, and that's where the next one gets added. So if we're talking about 5' to 3' in the forward direction, you can sort of think of it as like puzzle pieces that are going to be joined together. Now, what does this mean though, 5' to 3'? Where did that come from? And 5' and 3', they're talking about the carbons. They're simply numbering the carbons that are found in the sugar. So again, here we've zoomed in. Here's our phosphate group. Here's our deoxyribose of the nucleotide. And here's adenine. In this particular case is the base. Now here in adenine, they number the different atoms as well. 1, 2, 3, 4, 5, 6, etc. And to differentiate those, to tell apart the atoms you're talking about in the base versus the atoms you're talking about in the sugar, instead of just numbering the carbons in the sugar again, they numbered them, but then they put a prime after it. It kind of looks like an apostrophe, although it's a little bit different. So now we can number the carbons in the sugar, and here they are. Here's the first carbon, so we call that 1'. Second carbon, 2', 3', 4', and 5'. So when we say 5' to 3', we're sort of just indicating where the next nucleotide is going to be bonded. At which carbon in the sugar does the next phosphate group get bonded? So let's take a look at this, right? Okay, so here are 5 carbons, 1, 2, 3, 4, 5. And we know that the next nucleotide is, we can just see the little part of the phosphate group. The next nucleotide's phosphate group bonds to this carbon, and that's the third carbon, the 3' carbon. So remember, it goes sort of in this direction from top to bottom because we have phosphate group, sugar, phosphate group, sugar, phosphate group, sugar. So the phosphate group is on the 5' end, and the next nucleotide gets added to the 3' end. And so the direction we're going here is from top to bottom, 5' to 3', right? In the direction of the 5' carbon to the 3' carbon. And then the next nucleotide gets added after that. So in our forward direction, the phosphate group is attached to the 5' carbon. Then we have the sugar, right? And at the 3' carbon of the sugar is where the next nucleotide gets added. So we're always adding new nucleotides to that 3' carbon of the sugar. So phosphate group, sugar, base. And then we bond the next phosphate group of the next nucleotide to the 3' carbon, right? Phosphate group. Now then we have the next sugar, and then the next phosphate group is going to be bonded to the 3' carbon. Okay, so we're really building from top to bottom, just like we had with the example that I drew out of the nucleotides. And that direction then, if you put it all together, building from top to bottom, here, 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 you're going in the direction from the 5' carbon at the top, going towards the 3' carbon. That's where the next nucleotide gets added. And now we've got the next sugar. And at the 3', that's where the next one gets added. So we're building in the 5' to 3' direction. And that's why we call the forward direction 5' to 3'. It's really just showing in that sugar of our nucleotide which carbon has our phosphate group that's part of this one, and then which carbon is going to bond to the next nucleotide. Okay, 5' is at the top, 3' is at the bottom, and that 3' carbon is where the next nucleotide gets added. So again, you can kind of think of these as like puzzle pieces, okay? And I know this is vastly simplified, but here's our first nucleotide. The 5' end is at the top, 3' ends at the bottom. Now we're going to add the next nucleotide, right? 5' ends at the top of the next one, 3' at the bottom. And we'll add the next one and the next one. And we can go on from there. So if we look, we started at the 5' end at the top, and then we go from 5' downward towards the 3'. And it's always going to be that 3' end that's sort of at the leading edge, right? That's where the next nucleotide is going to be added, always to the 3' end. So we're going from 5' to 3'. 5' is the first one that gets added, and then every next nucleotide gets added onto the 3' end of the nucleotide that's there before. So if we look back at this graphic and we see, okay, here's one nucleotide, here's the next nucleotide, here's the third nucleotide, and so forth, I want you to take a look at this. Which is the 5' end of that strand, and which is the 3' end of that strand? Okay. So maybe this is what you said, that the 5' end is on this end, and it's going from top to bottom. It's going from 5' to 3'. That's the forward direction, because each new nucleotide was added to the 3' end. So here's our first nucleotide. And then at the 3' carbon, that's where the next nucleotide gets added. And then the next nucleotide. So we're going from top to bottom in this case, from 5' to 3'. Now let's take a look at the other strand. Which direction is the other strand going? Okay, well, we know the DNA is antiparallel. So if the left strand is going from top to bottom, then we know that the right strand in our diagram is going from bottom to top. And so bottom to top, that's the forward direction. And we know the forward direction is 5' to 3'. So 5' then must be at the bottom. Okay, you can pause it here and take a look and see if you understand this, or maybe even if you can explain it to somebody else. Because if you can explain it to somebody, that shows you which pieces you're missing in your understanding, or if you've got it all down. So again, 5' to 3' is the forward direction. We use this notation just because the nucleotides here get added to the 3' end. So the 5' end is sort of the leading, okay, is the first part, and it goes from 5' to 3'. And on the 3' end, that's where the next one is added, okay, 5' to 3'. And then the next nucleotide gets added. Here's this 5' end, here's its 3' end, and that's where the next nucleotide gets added. So we're building from top to bottom, 5' to 3'. And since the other strand is antiparallel, then it's going in the opposite direction. The 5' end is on the bottom, 3' end is on the top. And that's why it's upside down. You know, you can see where the oxygen is found. It's sort of at the top of that pentagon of the deoxyribose sugar. And so here, if we're going from top to bottom, the oxygens are at the top. But here, when we're going in the opposite direction from bottom to top, we can see, well, this whole strand is in the reverse order. The oxygens are actually on the bottom. And then the different bases, adenine, thymine, cytosine, or guanine, they stick into the middle of our double helix structure, and they base pair with each other. So adenine is always going to base pair with thymine, and cytosine is always going to base pair with guanine. And again, if it's of interest to you, the nucleotides are bonded together through what we call phosphodiester bonds. And those are covalent bonds. They're very strong. But the bases are bonded together through relatively weak bonds called hydrogen bonds. And these are bonds that can be broken. So there are situations like in DNA replication where the two strands of DNA have to separate from each other for replication to happen. And so those hydrogen bonds actually get broken and the two strands separate. So this is what we mean when we talk about 5' to 3'. That's the forward direction of DNA. And if you don't have to, or you don't want to remember the reason why we call it 5' to 3', then just remember that 5' to 3' is the forward direction. And that's always the forward direction. That's how DNA is going to be made. New DNA is going to be made from 5' to 3'. Messenger RNA is also going to be assembled from 5' to 3'. Okay, so it's always in the forward direction and never backwards. So here we go. Here's our DNA, our double helix. This strand, if this is the 5' end, right? 5' to 3'. This is the other end. It's going in that direction. Now, the other strand goes in the opposite direction. So here it is going forward from 5' to 3'. That's the forward direction of our DNA. And there are 4 different bases. They can fit into the center and form what we call base pairs, right? Cytosine, guanine, adenine, and thymine. Those are the 4 different nitrogenous bases that are going to stick into the middle of the DNA molecule and form those base pairs. One of them is on one strand, right? A nucleotide on one strand, and the other base is part of a nucleotide on the other strand of the DNA. And that's our third rule about assembling DNA, is that adenine on one strand of DNA is always going to pair with thymine on the other strand. And the same is true for cytosine on one strand and guanine on the other strand. We call this complementary base pairing. Now, this isn't compliment like someone said something nice about you, right? That's like a compliment, and that's actually spelled different. That's with an I, right? That's where the I is, P-L-I, compliment. They paid you a compliment. This is compliment with an E. And when you use the word complement with an E, it actually means you create a whole, that you fill in something that isn't you. In other words, like if 2 people really complement each other well, that means that together they really have different skills. So if this person's really good at this, maybe this person is really good at something else. And together they complement each other. They bring something different into the whole. And you can sort of think about this because this word complement, it's the same root as complete. So, you have 2 things that complete each other. And adenine and thymine are going to bond to each other. They have a complementary base pair with each other because they sort of complete each other that way. One is a pyrimidine and one is a purine. So they complement each other. So we call those complementary base pairing. So here, when we look at the different bases of DNA, here in orange is guanine. So we know guanine is always going to form a base pair with cytosine, which is in red. And then the purple one here is thymine. So we know on one strand, if it's thymine, it's going to be adenine on the other strand, okay? So T and A always form a complementary base pair. And then the other 2, C and G, always form a complementary base pair with each other. And it doesn't matter if guanine's on this strand, or if it happens to be on the other strand, okay? The other nucleotide on the other strand is always going to be cytosine. And the same is true if thymine is on this strand, well, it's going to be adenine on the other strand of DNA. Or if adenine is on this first strand, then it's going to be thymine that forms that complementary base pair on the other strand. Now, this complementary base pairing is really important to keep in mind because when we're copying DNA or when we're creating RNA, it's going to follow those same rules, where if new DNA is being laid down in the 5' to 3' direction always, well, those base pairs in the nucleotides are going to be complementary to the template of DNA that's being used to copy from. Okay, so we'll talk about that in a different video if you're interested. So here's a question for you, something to try out. Here we have just a small piece of DNA, and we know it's going from left to right because there's the 5' end on the left and the 3' end on the other side, right? So we know this piece is going left to right. Now, our sequence of bases in these nucleotides is ATTGACT-CAC. Okay, so adenine, then thymine, guanine, adenine, cytosine, thymine, cytosine, adenine, guanine. That's the order of our bases of the nucleotides from 5' to 3'. So my question for you is, can you determine what the other strand of DNA, because DNA is double-stranded, what will be the bases that correspond to this strand of DNA? Okay, so I'm going to give you a second, but see if you can figure out, we're trying to make the other strand. And just by having this one strand, we can determine what the base sequence is in the other strand by using complementary base pairing. Okay, I'm going to give you a second, see if you can figure it out. And remember, you can always pause the video if you need more time, okay? All right, you ready to walk through this? Okay, well, if on this strand is a nucleotide with an adenine base, then we know adenine forms a complementary base pair with thymine on the other strand. So the other strand is going to have a thymine bonded there. Okay, the next nucleotide in our sequence here has a thymine. In fact, we have adenine and then two thymines. So we know in the other strand, we're going to have thymine and then adenine and adenine, right? Because if on one strand we have thymine, we know we have adenine on the other strand. Now here we get to a guanine. So this is a little different. Guanine on this strand means that on the other strand, we're going to have a cytosine, right? Because guanine and cytosine always form that complementary base pair. So on this other strand, we're going to have T, A, C, T, G, A, G, T, C. That's going to be the sequence of our bases in the nucleotides of the other strand. And we know this because of how these biochemical and physical rules work within DNA, where if there's adenine on one strand, then that means there's going to be a thymine base pairing with it on the other strand. So thymine is going to pair with adenine. Here's a thymine, so there's going to be adenine in this one. Same thing here. There's a guanine on this strand. There's going to be cytosine on the other strand, and so forth and so on. So you can sort of test yourself by coming up with some random arrangements of bases in a strand of DNA, and then see if you can come up with the complementary base pairing, which nucleotides are going to be on the other strand and which bases are going to be found in those nucleotides. Okay, now here's my last question for you. Which side of this new piece that we made is the 5' end, and which is the 3' end? Remember, 5' to 3' is going to be in the forward direction. So if this side here of our old strand is 5' and the right side is 3', we know that this piece is going from left to right. Now, we also know that DNA is antiparallel, which means that the other strand goes in the opposite direction. So if this top strand is 5' to 3', left to right, then it makes sense that the new strand is 5' to 3', right to left. Okay, so our top strand is going left to right. Our bottom strand is going right to left. And we know that forward direction, is 5' to 3'. So 5' to 3' is the direction on the top strand. It's going left to right. The bottom strand, therefore, is going right to left, which means that the 5' end is on the right side and the 3' end is on the left side. Okay, so they're anti-parallel. They go in opposite directions. And again, why do we care about this? Well, because new DNA is always going to be made in the 5' to 3' direction, and it works anti-parallel on its template. So when we're learning about how DNA is replicated or messenger RNA is made, we need to always know, are we talking about the 5' end or the 3' end? Okay, which direction is this strand of DNA going? Okay, so bringing it all together, we know that to assemble DNA, that DNA is composed of two strands of nucleotides. Okay, the nucleic acid, deoxyribonucleic acid, is made up of nucleotides bonded together. And not just one strand of nucleotides, but two strands of nucleotides. Now, they are parallel to each other, except one's going one direction, one's going the opposite direction. So they're actually anti-parallel. Okay, that's what we call it, anti-parallel. When one strand's going one direction, the other strand is going in the opposite direction. So they're still at a set distance apart from each other, but they're going in opposite directions. Now, complementary base pairing rules tell us that whenever we have an adenine in one strand on this nucleotide, in the other strand's nucleotide, well, that adenine is going to pair with a thymine base in the other nucleotide. And so A and T always base pair together, and C and G always base pair together. And we call those complementary base pairing rules. Okay, so let's think about what we learned today, the structure of DNA. And I know that was a lot. So maybe you want to go through the different parts individually just to review to make sure you have a good understanding. But we learned what is a nucleotide of DNA, how do nucleotides relate to nucleic acids, which nucleotides are found in DNA, and what do 5' and 3' mean. Now, if you want, you can pause the video here and see if you can answer each of these questions. And then after you've paused the video, you can unpause it and come back, and I'll go ahead and explain the answers to all these questions. See if we can come up with sort of a concise answer to all of these. Okay, did you pause the video? Okay, well, let's talk about the answers to these now, okay? What is a nucleotide of DNA? All right, well, a nucleotide is just the building block. It's these smaller structures that are joined together to form the larger nucleic acid. So just like a whole train is made up of all these different railroad cars, the nucleic acid, DNA, deoxyribonucleic acid, is made up of lots of different nucleotides that are bonded together, attached together to each other, okay? So nucleic acids are made up of these smaller structures bonded together called nucleotides. So the second question is, how do nucleotides relate to nucleic acids? Okay, well, I guess I just answered that. The nucleotides are going to be bonded together, right? So to create longer structures that are then known as nucleic acids. And remember, as part of the first question, maybe we also wanted to say, if this was like a short answer question, an essay question on an exam, maybe what is a nucleotide of DNA? It might be good to talk about the structure of a nucleotide, where we know in a nucleotide, there's a phosphate group, and that phosphate group then is bonded to a sugar. It has 5 carbons in it, that sugar, and that sugar is called deoxyribose. And then on the other side of that deoxyribose is where that nitrogenous base is attached. And there are 4 different types of nitrogenous bases. And that sort of is part of the answer to question 3. Which nucleotides are found in DNA? Well, we have 4 different options, right? Each of them is going to have a phosphate group attached to deoxyribose, the sugar. But then the base that's found can be adenine, it can be guanine, it can be cytosine, or it can be thymine. So we actually have 4 different types of nucleotides because in DNA, we have 4 different types of nitrogenous bases that can be in there. And then in the fourth question, what do 5' and 3' mean? Well, officially, we're actually talking about the different carbons that are in the sugar of the nucleotide, which is deoxyribose. So we number those 1', 2', 3', 4', 5'. So 5' and 3' are referring to the sugars. But more importantly, when we use the word 5' and 3', we're talking about the forward direction of DNA. That is how the nucleotides are joined together, right? And they're always joined together from the 5' end to the 3' end. So the forward direction of DNA is from 5' to 3'. 5' to 3', 5' to 3' is the forward direction of the DNA. So if we're making DNA and from these nucleotides, joining them together, 5' is going to be the first nucleotide on the 5' end, and then new nucleotides are always going to be added to the 3' end. It also helps us keep track because we know that the 2 strands of DNA are anti-parallel. So if this one's going in this direction from 5' to 3', we know that the 5' and 3' direction of the other one is in the opposite direction. Okay, so we talked about the structure of DNA today. We talked about how all these nucleotides come together. And now it's important to sort of go through and review and see if you can explain and draw out these principles on your own. Thanks for watching. And if you found this at all of use, please like it and subscribe to my page, and that will help other people find this information as well.